The presence of microorganisms in the brain is a topic of growing interest and research. It has been traditionally believed that the brain is a sterile environment, protected by the blood-brain barrier which prevents the entry of harmful substances and pathogens. However, recent studies suggest that there might be a microbiome within the healthy brain, similar to other parts of the body like the gut and skin. Researchers have found evidence of bacteria and other microorganisms in the brains of both healthy individuals and those with neurological diseases. For example, a study found non-human genetic material in brain tissue, indicating the presence of bacteria and phages in the brains they studied. Interestingly, the human brain microbiome appears to be a subset of the gut microbiome, with about 20% similarity. In healthy brains, the microbes do not seem to cause inflammation or bacterial disease, suggesting they may reside harmlessly. They tend to cluster in specific brain areas such as the hippocampus, prefrontal cortex, and substantia nigra. This discovery opens up new avenues for understanding the role of these microorganisms and their potential impact on brain health and disease. It's important to note that while these findings are intriguing, the research is still in its early stages. The exact mechanisms of how these microorganisms enter the brain and their functions within it are not yet fully understood. Further research is needed to map out the complete microbiome of the brain and to understand the implications for neurological health and diseases like Alzheimer's. Let's try to design a possible research experiment to examine the function of microorganisms dwelling in healthy human brain. To examine the function of microorganisms dwelling in healthy human brains, a well-designed research experiment is crucial. Here's a proposed experimental design. Objective: investigate the presence and potential functions of microorganisms in healthy human brains across different age groups. Participants: recruit a diverse group of healthy individuals across various age ranges, e.g., 20-30, 30-40, 40-50, 50-60, 60-70 years old, with no history of neurological disorders or recent antibiotic use. Obtain informed consent and relevant medical history. Sample collection. 1. Collect brain tissue samples from consenting participants undergoing elective neurosurgical procedures, e.g., tumor resection, epilepsy surgery, or post-mortem brain donations. 2. Implement strict aseptic techniques and contamination controls during sample collection and processing. Experimental groups. 1. Healthy brain tissue samples from different age groups. 2. Negative control. Sterile brain tissue samples, if available, or samples processed with DNA, RNA decontamination protocols. Methodology. 1. Extract and purify DNA and RNA from brain tissue samples using appropriate techniques to minimize contamination. 2. Perform high-throughput sequencing, e.g., 16S rRNA gene sequencing, metagenomic sequencing, to identify and characterize the microbial communities present in the brain samples. 3. Analyze the sequencing data to determine the diversity, abundance, and taxonomic composition of microorganisms in each sample. 4. Compare the microbial profiles across different age groups and with the negative control samples. 5. Perform functional analyses, e.g., metatranscriptomics, metabolomics, to investigate the potential roles and activities of the identified microorganisms in the brain. Data analysis. 1. Use appropriate statistical methods to analyze the microbial community data, accounting for potential confounding factors, e.g., age, sex, diet, environmental exposures. 2. Perform correlation analyses to identify associations between microbial profiles and age, as well as other relevant factors. 3. Employ bioinformatics tools and databases to predict the functional capabilities of the identified microorganisms based on their genomic and transcriptomic data. Validation. 1. Confirm the presence and viability of identified microorganisms using culture-based techniques or fluorescence in situ hybridization, FISH, on a subset of samples. 2. Perform additional experiments, e.g., germ-free animal models, in vitro co-culture studies, to validate the functional roles of the identified microorganisms in the brain. This proposed experimental design aims to comprehensively investigate the presence, diversity, and potential functions of microorganisms in healthy human brains across different age groups. Rigorous contamination controls, multi-omics approaches, and validation techniques are essential to ensure the reliability and reproducibility of the findings,